In agricultural systems, animal urine is the main contributor of nitrous oxide emissions. The production and consumption of this gas is regulated by microbial communities through nitrogen transformations. In soils exposed to urea, a community succession is triggered driven by the replacement of organisms. However, the mechanisms driving these replacements are unknown. Here, we expose soil microcosms to urea to trigger a community succession. We added biological inhibitors to determine the role of competition in mediating succession. This plot shows community composition. The shapes indicate the different treatments and the color indicates time. Closer points indicate similar composition. We see here that two separate clusters are formed between the no urea and urea treated samples. This indicates that urea shifts community composition. The plot on the left shows species richness, which is the number of unique organisms in a community. The black line represents no urea, the red line represents urea only, and the bars represent urea and inhibitor treatments. We see here that urea decreases species richness, while inhibitors induce the recovery of species richness. A similar trend is seen across the taxonomic level, where more number of taxa are seen without urea compared to when urea was added. This indicates that urea favors specific groups of organisms. When inhibitors were added, a larger number of taxa was present. Altogether, this shows that inhibitors induce the recovery by removing competition through the suppression of actively growing organisms. In conclusion, we show that community succession is triggered in response to urea, that competition was driving community succession, and that the mechanisms mediating succession was through the inhibition of actively growing organisms.